Today, I'd like to walk you through how we visualize somatic point mutations from spatial transcriptomics data. And I'm assuming you have some familiarity with the software Integrative Genomics Viewer. Here I'm showing you a snippet of DNA and RNA sequencing reads from a gene called UBXN1. This top track corresponds to DNA sequencing reads from a cutaneous squamous cell carcinoma. And the second track corresponds to DNA sequencing reads from a patient-matched reference. So this is non-tumor tissue. Now, any sequencing variants in the tumor, but not the normal, these would be somatic point mutations. And any variant reads that are in both, those would be germline polymorphisms. So immediately you might notice that there are variant reads shown here in both the tumor and the normal. This would be a germline polymorphism. But more interestingly, at least for a cancer biologist, you also will see some variant reads here in the tumor that are not in the normal. And this corresponds to a somatic mutation that was called using the MUTECT2 algorithm. This particular somatic mutation is a CC to TT mutation. I can zoom in to show you, which is a very common mutation type in UV radiation induced cancers. Now, this third track, I'll zoom out again, corresponds to the RNA sequencing reads from the spatial transcriptomics data. As you could see, the coverage is biased towards the 3' end. So the 5' end of UBXN1 is over here, and the 3' end is over here. Transcription goes from, in this case, right to left. The DNA sequencing reads tend to be enriched over the exons, and that's because exome sequencing was performed, whereas the RNA sequencing reads are enriched towards the 3' end, and that has to do with the chemistry that the 10x Visium platform utilizes to perform spatial transcriptomics. This particular track corresponds to all of the RNA sequencing reads from all of the individual spots on the Visium array. As you can see at its highest coverage, there's roughly 375x coverage from the RNA sequencing, and then this tapers off as you move closer to the 5' end of the gene. And then lastly, this final track corresponds to individual sequencing reads from one of the spots on the Visium array. So let's zoom into our mutation. As again, this is a T to T or a CC to TT mutation, and you can see quite a few reads in the tumor DNA. You will not see any reads in the normal DNA. And then in the spatial transcriptomics data, you will also periodically see a read. Now, interestingly, whenever there is a mutant read, you'll notice that there's improper splicing. And so this suggests that not only is this mutation expressed, but when it is expressed, it disrupts this predicted splice site and results in a read-through of this intron. Now, in the spatial transcriptomics data, you can hover over a read and you'll see all of the various tags from the BAM file. And the CR tag corresponds to the spatial barcode, and the UR tag corresponds to the unique molecular identifier. So 10x Genomics has software that allows you to parse this BAM, which includes reads from all spots, into individual BAMs that only have reads from each spot. And so that's shown here. Now, in total, there were 19 mutant reads from all spots, and this particular spot has one mutant read and one reference read. So it's not surprising given that there were nearly 3,000 spots with tissue underneath them and only around 20 spots of mutant reads that we would only find one mutant read from a given spot. It's also not surprising that we would see both a mutant and a reference read, and that is because this mutation is probably heterozygous, meaning that we are sampling from each allele. Now, as the next step, once we've identified the spots that have mutant reads, we import those spots into the Loop browser and visualize where they reside within the tumor. And so that's shown here, where we imported spots with 2 or more mutant reads, 1 mutant read, and then various combinations of reference reads, so 0 mutant reads and 1, 2, 3, 4, or 5 mutant reads. And the idea here is that we believe a spot with 2 mutant reads is almost certainly overlying tumor tissue. A spot with 1 mutant read is also likely to overlie tumor tissue. The only way that this could occur by random chance would be if there was some sort of a sequencing error that happened to result in the detection of a mutant read. Whereas we used a higher threshold to call the non-tumor spots. And again, that's because if a somatic mutation is heterozygous and you only have 1 or 2 reads mapping to the reference allele, that could be a sampling bias where the wild type allele was, for whatever reason, sequenced preferentially over the mutant allele. But once you get to 5 or more reference reads, we feel like the likelihood that there are tumor cells underlying the spot decreases substantially. So when you visualize where these spots are located, you can see from this particular tumor, the majority of those tumor-bearing spots do correspond to the regions that the pathologist marked as histologically likely to be tumor tissue. And then this particular case generally had lower spatial transcriptomics sequencing coverage, but you can still see what appears to be mostly normal epithelia up here, and then the region with cutaneous squamous cell carcinoma does have some scattered spots with mutant reads. So this is a general overview of how we visualize somatic point mutations in Visium data. Now my colleague Li-Ming Chen is going to walk you through the scripts that allow us to carry out each one of these steps.
Today, I'm going to walk you through an R package called STMute, which we developed to explore 10x spatial transcriptomic data. This package covers analysis of point mutations, allelic imbalance, and copy number variation. The installing of this package is easy. You just copy-paste these 2 lines of code in your RStudio, then you will be all set. Before using this package, make sure you have sequenced your DNA data and generate bulk CMV, germline, SNPs, and a somatic mutation list. Also, you need to make sure you have run the Space Ranger pipeline successfully, because there are 5 files we will need for later data analysis. I listed here of these 5 files. The second one, graph-based CSV, you need to export from Rupe browser by clicking the 3 white dots, and then click Export Graph-based. This graph-based CSV will be automatically saved in your computer. Now we have all the data ready. Let's get started with point mutation analysis. The first function you need to run is split spot. It takes 1 argument, filtered feature barcode CSV, and it will generate a folder called spatial in your working directory. Inside this folder, there will be 2 subfolders called spot index and txt. In the spot index folder, each barcode is saved into an individual txt file, and it is given a specific spot name. And inside txt, it contains a list of spot gene expression profile. Of this spot gene expression profile, it has 2 columns. The first column is a list of genes. The second column is the RNA-seq data. Next, we need to generate a BAM file for each spot. Here we suggest 10x genomic subset BAM. We provide our code here for your reference. Next, we also need to count the number of ref and mutant reads for each spot. We provide our customized Perl script, which you can download here. And then here is the code we use for your reference. In the end, you just need to run spot point mutation function. This function takes 6 inputs, which we listed here. And it will generate 1 folder called point mutation in your working directory. It contains 8 files. The first one is the one that can be visualized in Rupe browser. And you are able to get an image similar to figure 1 in our manuscript showing here. You are able to see which spots have the most mutant reads and in which part of the tissue that they are located. Another file that will be interesting to you is this one. Inside this file, you are able to see exactly what kind of mutation each spot has. To get more information of the meaning of each column, please refer to our GitHub page. Next, I will talk about the copy number variation.